Okay, can everybody see that?
Yes.
Yeah. Okay, so, well, thank you very much, Carol. I'd just like to thank you and the Pender Harbour Wildlife Society for inviting me to give this talk this evening about the amazing world beneath our feet and why it's so important for our whole existence, because these organisms in the soil are recycling nutrients, and without them doing this, we'd be buried knee deep in litter and waste. And they're also really, really vital in terms of forming and retaining soil organic matter, which of course is really important for soil carbon sequestration, which is vital now in these times of climate change. So what I wanted to do is to start off by talking about what is in the soil, for those of you not so familiar with those organisms, and what they do in the soil, why they're so important, what we know about them. And then I'd also like to talk about the vital role that living roots play in feeding those organisms, and why, because of that, we need to really start rethinking about how, for example, we manage our forests, because the present practice of clear cutting is just not sustainable. And as Carol mentioned, the article from National Geographic that was in the September issue, I just wanted to start by acknowledging Oliver Mex and Nicole Ottawa, the photographers who took the pictures, and they've allowed me to use those photographs in my presentations. So just an acknowledgement to them for the pictures I'm going to be showing. So I wanted to start off by talking about soil biodiversity. So you may or may not know that soil probably harbors most of this planet's undiscovered biodiversity. So for many, many years, people have thought soil is just an inert substance that covers the surface of the earth, but it's teeming with organisms. And so if you took a tablespoon of soil, there's more microbes in that tablespoon of soil than there are people on the earth. And there's thousands of species. So in that same tablespoon, you'd find 1,000 fungal species, up to 50,000 bacteria, probably 4,000 archaea, and 89 nematodes, which are the small soil animals, and 159 mites, which are another type of soil animal. And we know this because we know the numbers of organisms in soil because we can look at them under the microscope, but we can't identify them because a lot of them look the same. So it's not the same as studying above ground plant diversity animals. You can't identify microbes by morphology. So it's only in the last 20 years since molecular biology has come on stream, and we can now extract DNA from soil, and then using a kit, we can extract that DNA in 4 hours, whereas my first postdoc, it took him 9 months to actually develop a method in the 1980s to extract DNA from soil. But that's how we know there's this many species, different species in the soil now. So when I was first at university, we had to try and culture organisms from soil, so grow them on agar plates. And we now know that organisms we have in culture represent less than 1% of organisms that are in existence. And I really like this quote, which is as apt today as when it was first quoted. that we know more about the movement of celestial bodies than about the soil underfoot. And this quote was actually made 500 years ago by Leonardo da Vinci. But I always say to the students, you know, you talk about space being the final frontier, but it's actually soil where it's all happening. So why is soil biodiversity important? Well, as I mentioned, they're really important in recycling nutrients. So plant litter material, animal waste, they're key to energy flow and storage in soil. And David Jenkinson was a soil microbiologist at Rothamsted in the UK, and he really started a whole new paradigm in soil science when he developed a method to extract microbial biomass and to measure it in soil. And that really changed in the 1970s people's view of soil being a living ecosystem. So they're really important for recycling nutrients, for generating soil organic matter. We now know that most of the carbon stored in soils is actually a microbial product, and it's very recalcitrant, really important for carbon storage. These soil organisms produce and consume gases that affect global climate. So for example, there's microbes that can oxidize methane in the atmosphere, removing that really toxic greenhouse gas produced a lot in agriculture. They can oxidize methane to produce the less potent CO2 gas. Microbes are used to destroy pollutants. So a lot of the hydrocarbons, you know, if we get oil spills, microbes are used to clean up those messes. Microbes are the basis of our sewage treatment works. And increasingly, microorganisms are being used as biocontrol agents against pathogens and insect pests. And this is seen as a green chemistry alternative to using harmful chemicals and herbicides. And so microorganisms were really our first source of antibiotics. So Penicillium notatum was where we got penicillin from. In the first place, it's a soil fungus. And biodiversity is really important because more diverse communities are more resilient to any perturbation we do to the soil. So I wanted to start off by just showing you this nice illustration, which was actually made by a colleague of Cindy and mine at UBC for a review in this area that we've just written. And it gives a nice overview of what's happening, some of these interactions in the soil, and how important, as I say, living roots are for providing these organisms with fuel below ground. So we now know that plants exude, so they leak out from their root systems about 50% of the carbon that they fix through photosynthesis. And 90% of plant roots have these mycorrhizal associations. So this is a mutualistic symbiosis between plant roots and fungal species. And this is a benefit to both partners. So the fungus is supplied with carbon for growth from the plant. And in turn, plants that have mycorrhizae on the root systems have greater access to nutrients and water. Because if you think about the surface area in the soil that these hyphae are exploring, it's like having a major new root system. And what I wanted to show you in these enlarged bubbles is some typical interactions you might see in soil. So here in this first bubble, we have a plant feeding nematode. And we know the nematode worms, these tiny worms in soil that you can just visualize with a microscope. They have different ways of getting their food sources. So this is a plant feeding nematode, which we can identify because it has a stylet, almost like a needle, which pierces plant roots, and it sucks out the carbon it needs for growth from the plant. And then in the wings here, this yellow organism, that's a collembola, a predatory collembola, which is waiting to eat that nematode. In this next enlargement, we've got some mycorrhizal roots here in purple, and then there's some smaller pink collembola that are grazing on the fungal hyphae on those roots. And then here in red, we have a predatory mite, like the red velvet mites you may see in soil. They're tiny, but you can identify them again using microscopy, and that's waiting to eat those collembola. And then in this last schematic, we've got the plant roots again and the mycorrhizal hyphae, and they're covered in these rod-shaped bacteria. So those bacteria are feeding on the exudates, so the sugars, the amino acids that are being released from the plant roots, the bacteria eat them, and then those bacteria in turn are eaten by single-celled protozoa. So these tiny small animals engulf the bacteria and get their nutrients from the bacteria. And what you see here are some of the larger soil fauna, things like centipedes, beetles, pseudoscorpions, and these prey on the smaller soil fauna. And I just wanted to, on the right here, give you an idea of the numbers of these organisms that you may see. This is in a teaspoon of soil. So you have 10 billion bacteria, 200,000 fungi, maybe 5,000 protozoa, 100 of the mesofauna. So these are the fauna that you really need a microscope to look at. And then if you took a teaspoon, you'd have part of an earthworm and part of these larger soil fauna. So this is what we call a typical pyramid, food web pyramid. So the larger organisms eating the smaller ones in the soil. So I'm just going to start to show some of the photographs of these organisms, and these first ones are the macrofauna that you're probably familiar with if you do a lot of gardening. So we call these macrofauna that ingest litter material in the soil ecosystem engineers, because they produce what we call biogenic structures. So things like earthworm casts and ant hills, so ant nests, where ants take and redistribute litter on the surface of the soil. And these casts as well, that after the worms have ingested litter material, they'll poop out these casts. And these are all areas of really intense high microbial activity. Some of you are probably familiar walking around the forests here on the coast with these yellow spotted millipedes, also referred to as night train. So these are our dominant litter transformer in our forest soils. They really like to eat big leaf maple leaves and also conifer needles. And then again, they poop them out into the soil. And again, these are hotspots of microbial activity. And then we have the termites or isopods, and these eat woody material. They can't digest them themselves, but they have bacteria in their guts that can decompose the cellulose in wood and break it down. We also have the very important dung beetles in soil, and these have a great diversity again in the soil. So we have beetles that are what we call dwellers. So they actually live in dung in the soil and will plant their eggs in that dung. Then we have this middle yellow and black species here, which is a tunneler. So it takes dung deep into the soil, redistributing that organic matter. And then we also have the rollers, this final beetle here on the right, and they take dung not very deep into the soil. And again, they'll lay their eggs in the dung, and then the larvae can feed on that dung when they hatch. So really important litter transformers, these macrofauna. And then we have the predatory macrofauna in soil. So things like this copper centipede here, you may be familiar with. They scuttle across the soil very fast looking for their prey. We have spiders, beetles, and ants, and these are all predatory on other smaller fauna. And then we come to the mesofauna. So these are some of the photographs, as I say, that were in the magazine that I have permission to use for the talk. And when people, and when I talk to students about the fascination with soil biodiversity, and I'll say, well, they're not cute and cuddly like bears. But I mean, what's not cute and cuddly about this water bear? It's a tardigrade. It looks so cute, and it's got 8 legs and little claws here. And it uses its mouth part to suck in litter material and detritus. It'll also eat nematodes. So it lives in moss and particularly wet forest soils. And it's really resistant to like really harsh conditions. So it can live in really cold conditions and really hot. And it can dehydrate its body to become inactive. And then when conditions are suitable, it can rehydrate and then become active again. So really interesting animals. And you can see how small it is. This is magnified almost 2,500 times. And then in this picture, we see more mesofauna. So again, if you're looking at the soil, you could probably see just a tiny, they're probably pinhead size, these mites. But as I say, you may have seen the red velvet mite because it's so bright red. But in this picture, we have 2 good examples. We've got a bristleworm here, and then an example of an oribatid mite. So these oribatids they live more on fungi and bacteria, whereas this mite over here is a mesostigmatid mite, and that's predatory on other fauna. So a much more scary looking creature in the soil. So these are really important again in the soil. And then we have these cute creatures. These are the collembola or springtail. And they grow to less than 1/5 of an inch. And their name comes from this appendage at the end of the body, which allows them to spring sort of 20 times their body length in the soil. And some of these are predatory. As I've said, they'll prey on nematodes in soil. Some are also bacterial and fungal feeders. And again, if you were to go out in forest soil and take a lump of that forest floor, and we do hand sorting, looking at that soil, and you'll quite often see collembola jumping. So you can visibly see them with the naked eye, but again, microscopy is great for looking at how these organisms look. And then this is a really nice example of hairy back, which are microscopic animals in the phylum Gastrotricha, and they live in the thin water films that permeate between soil aggregates. And they move through the earth using these hair-like cilia you can see on the surface in search of bacteria and algae to eat. And then this is a rotifer. So this is a rather spiky-bodied animal. And again, it lives in the water films in soil, and they propel themselves using these hair-like fibers on their surface and ingest organic debris as they float through the soil. And then we have the nematodes, which are the microscopic worms. And they, as I mentioned, have very diverse feeding habits. So some of them feed on plants. So they have this stylet that they use to inject into the plant root and suck out carbon. Others are bacterial or fungal feeders, and yet others are insect pathogens. And the ones that are insect pathogens are now being considered for biocontrol of those insects. And then we come on to the other microfauna. So protozoa are considered microfauna, so the tiniest of the soil animals. And the protozoa also belong to the microfauna. And this is an example of an amoeba. And these protozoa have such a wide range of growth forms. And this one is, I think, pretty beautiful. It's a single cell parrot novella. And these are formidable predators. So what they do, they use contraction of their body to move through the soil, and they actually engulf microbes, so bacteria and fungi, and will digest them whole using enzymes. Another example of a protozoa, a testate amoeba, is this picture here. And this shell on this amoeba is made of silica. So it's thought that these hard shells that these animals possess protect them against environmental change in the soil. And you can see just how small these creatures are, because this is enhanced 14,000 times. And then this is another example of a protozoa. So these are the closest relatives to animals, but again, they're single-celled. They have these flagella on the surface, which help propel them through soil. And they also move the bacteria and prey and the detritus to this collar through which their prey is again engulfed and ingested and digested as a source of food in the soil. And then this very beautiful animal, these are known as the sun animals. And they feed again using these radial projections, the so-called axopodia, which enclose their prey organisms, which include ciliates and tardigrades and rotifers. And then again, they're digested in this food vacuole through a process that we call phagocytosis. And then some pictures here. These are actually slime molds. So this is a fruiting body of a slime mold releasing spores from its perch on this bit of woody debris, and there's also some fungal hyphae here. And this is another slime mold that has this characteristic white stalk and then a cap that's frequently brown and blue in color. And these slime molds were originally thought to be just a form of bacteria, but they're more similar to protozoa, to amoeba. And these slime molds feast on other microbes found in decaying plant matter. And you may be familiar with slime molds, such as you often see bright yellow or bright white slime molds on wood in the forest. And this is another good example. It's really apt name, the pretzel slime mold, Hemitricha serpula. So when there's a food shortage, what these slime molds do is to join together and migrate, so oozing across the surface of the soil, looking for food sources, so engulfing particles of food in kind of an amoeboid fashion. So getting bacteria and fungi and non-living organic matter as their food source. So pretty interesting organisms in the soil. And then, as well as the soil animals, we have soil microorganisms. And again, most of these are too small to be seen with the naked eye, but some of you may recognize some of the higher fungi that produce fruiting bodies. So things like the king bolete, also chanterelles, morels. There's other microorganisms in the soil like algae, yeasts, some of the lower fungi which don't produce fruiting bodies that you can see above the surface. We also have bacteria which can be rod-shaped or coccoid or spiral-shaped. These are archaea, which we also find in soil. And then you may be familiar with another type of symbiosis in forests. These are lichen. So this is a symbiosis between an algae, a fungus, and actually this Labaria lichen or lungwort lichen is a tripartite association. So it's a three-way symbiosis between a fungus, which gives the lichen its body, the algae, which is green within the fungus, and then we have a cyanobacteria, which is fixing nitrogen in this lichen. So these lichens are usually found on the branches of trees, but then they fall to the forest floor, and they're an important source of nitrogen in our forests. So using, again, electron microscopy, we can see some great pictures here of rod-shaped bacteria, some of which are in chains, some of which are single, some are in pairs, and that can tell you something about the species of bacteria, how they're growing. And these are magnified 2,000 times, so you can see this piece of wood. So the bacteria feed on exudates from plant roots, from mycorrhizal hyphae, and then this is an example of what we call a saprophytic fungus, so a fungus that is free living in the soil, but it's a wood degrader. And this is what we call a white rot fungus. So we call it white rot because if you see it degrading wood, when it's degraded that wood, you'll see white on the wood that it's been active on. And this is the cellulose that remains after it's broken down all the lignin in the wood. But what this species does is it's a successor to a different fungus, Phromitopsis, which is a brown rotter. And a brown rot fungi can only break down cellulose in wood. They can't break down lignin. So when you see decaying tree stumps that are brown, the cellulose has been broken down, but what's left is the brown lignin. So it's a nice example of succession of decomposition in forests. And this Antrodiella has really, it's able to outcompete the Fomitopsis once the Fomitopsis has broken down just the cellulose in wood. And it's actually quite a rare species in Europe. So as well as free-living fungi that we have in soil that are important in decomposition, we have these fungi that live in close association with plant roots. So they're mycorrhizae that you may have heard about. So as I mentioned, 90% of plants live in symbiosis with a fungal partner and called a mycorrhiza. So some mycorrhizal fungi called arbuscular mycorrhizae make their homes right inside the plant cortical cells. So this is the root of a grass species. And you can see this is fungal hyphae within those cortical cells. And so the fungi within the cells are receiving carbon from the plant. And in return, the fungus here is able to access organic forms of phosphorus by producing enzymes, which are then available for the plant to take that phosphorus up. So most herbaceous plant species, crop species, grasses, have this arbuscular mycorrhizae. And it's called arbuscular because these are arbuscules where the sites of exchange of carbon and nutrients occur within the plant cell. And then other types, so there are three main types of mycorrhizae. The arbuscular, another is ericoid. And ericoid mycorrhizae, similar to arbuscular, make their homes within the plant cortical cells in the root. But these ericoid mycorrhizae form associations with ericaceous plants. So things like heather, blueberry, huckleberry, salal, all have these associations. And these fungi are really, really good at producing enzymes to break down organic nitrogen in soil. So they're really good. And it's the reason why these plants can grow in really nutrient-poor soils, because they have these fungi associated with them that can break down organic nitrogen and release it to be plant available. And then the third main type of mycorrhizae that are formed with plants are those between ectomycorrhizae and tree roots in particular. And in this case, ectomycorrhizae, the mycorrhizae don't penetrate the root cells of the plant. So this is a scanning electron micrograph of a spruce root tip that's been cut through, and you can see these type of fungi form this really thick mantle on the fine roots, and they grow intercellularly, so around the cortical cells in the root, but they don't penetrate them. And these, because of this physical mantle, this thick mantle on the root tips, it's a physical protection from pathogens, and it also gives the trees greater access to nutrients and water in the soil. And I really like this picture. This is from David Reed in Sheffield, who's a great mycorrhizal expert. I worked with him during my PhD, and these are images of a plexiglass root box in which you've got a tiny conifer seedling, and the root just goes down. There's one main root here, But this is a really nice way to actually see what these mycorrhizal fungi do for the trees and the tree seedlings. As you can imagine, a big tree with all these mycorrhizal hyphae. So this is all the hyphae of the fungus. So it's like having a massive extension of your root system and surface area for getting nutrients and water. So when a plant is mycorrhizal, it benefits in several ways. It has greater resistance to plant pathogens because of that thick mantle. It has better nutrition, so better access to organic phosphorus and nitrogen, which abound in the soil and not plant available. Now, some of the fungi, there's thousands of species of mycorrhizae. Some of them will associate with many different tree species. So we call them broad host range fungi, things like Lacaria lacata here, or the king bolete, Boletus edulis. They will associate with many different trees, but then there are host-specific mycorrhizae, which are really interesting. Things like Suillus grevillei will only associate with larch trees. Rhizopogon vinicola will only associate with Douglas fir. And equally, some of the fungi are choosy which trees they associate with. Equally, the receptivity of different trees is different. So, for example, alder is known to only have low host receptivity. So it's only known to associate with 50 to 60 different mycorrhizae, whereas Douglas fir is known to associate with 2,000 different fungal mycorrhizae. The reason for this, we're still trying to understand why there are these so many associations, why they all be important. But if you went out into the forest and looked at the roots of a single Douglas fir tree, you're not going to find 2,000 fungi on that tree. You may find around 80 different species on the tree. And again, why? Well, we're trying to understand that. That's one of the big questions. So what do we know about biodiversity in the soil? As I said, it's only in the last 20 years that we've had the molecular techniques to start to try to understand about this diversity. And this just shows you what the current status is of our knowledge about animal genomes, plant genomes, and then bacteria in purple. In brown, we have the protists, the fauna, and we have fungi here in orange. And where we have these red on this outer ring, that's where the whole genome has been sequenced for that organism. So we have more whole genome sequences for things like bacteria, because of course their genomes are smaller than plants and animals. But in terms of the numbers of species we've named and what we estimate from the molecular techniques, so for bacteria, we're not doing very well. We've named around 7,000 out of an estimated billion in existence. For fungi, we're doing a bit better. We've named 100,000 out of an estimated close to 4 million species. And again, we're doing better with the higher organisms that can be identified by morphology. But hopefully, in the years to come, we'll be able to have named more of these species and know more about them and their importance in the soil. So we are, as well as identifying organisms based on their DNA sequences, because we can now know the genetic sequence of enzymes that are really important in, for example, mineralizing plant litter. So we can target, when we extract DNA from soil, we can target looking at that particular part of the DNA to see if it is present in the organisms that we've identified in that DNA from a different soil. So we're now, through these sequences, we're able to determine the potential of an organism, even if we haven't been able to culture it, its role in different processes in the soil. So for example, the pale yellow here, these are all different bacterial phyla, and we've now been able to show that particular phyla have the enzymes that are really important in terms of, for example, carbon mineralization, wood decomposition, nitrification, carbon fixation, nitrogen fixation. So we're beginning to build a picture from these molecular methods about what the potential is of these new organisms, even if we haven't been able to grow them yet. Now, I wanted to talk about, I've talked about the organisms in soil and how important the roots are for supplying carbon to these organisms. And like much of global biodiversity, terrestrial biodiversity is under threat from human activities such as agriculture and forestry. And this was actually put out by Interfor in celebration for National Forest Week in September. And this is just a really, it went viral on Twitter because it's just such a bad example. It's a good example of what industrial forestry is now, but it's not sustainable. If you look at that, we've got one living tree on that soil and all this debris. So we really need to rethink how we manage our forests to consider below ground biodiversity. So some of you may remember in the 1970s, 1980s, the public outcry over the clear cutting, particularly here on the coast. There was demonstrations over clear-cut sound in Haida Gwaii, which led to the ministry and the government developing alternative ways of harvesting rather than clear cutting. And so in the 1990s, variable retention harvesting was favored over these large clear cuts. So they were favored because of the visual perception, how it looked to the public. It was shown to maintain the biodiversity and function above ground, and also had benefits for subsequent tree regeneration. So there was two varying forms of retention. So what we call dispersed, which is basically thinning the stand at different amounts. So sort of 15% retention, 30%. And then we have aggregated or group retention with patches or corridors for wildlife to travel. So this was taken on in the 1990s, but we really haven't grown in terms of this variable retention, because what we're doing now is basically clear cutting with reserves, which is just not sustainable. Now we know how important the living roots are. So currently, the Forest Practices Code just recommends, in order to protect the soil after harvesting, the retention of coarse woody debris, which they consider to be roughly 8 hockey sticks left per hectare, and also limitation of soil erosion and compaction. But what we really need to be doing is considering retention of living green trees and not in massively cut areas. So in the early 2000s, we were involved in the STEMS experiment on Vancouver Island. So it stands for silvicultural treatments for ecosystem management in the Saywood. And the Saywood is the forest area north of Campbell River. And this is an aerial view of the treatments. So they had various ways of harvesting the forest. So the dispersed retention here, clear cut areas, and then some reserve areas, patch cuts. So we were interested in whether by leaving living trees on site, this would retain soil animals, soil microbes. And if they did, what pattern and size was required in order to keep the soil functional. So we were comparing aggregated, or these group retention, with dispersed, where you have single trees left dispersed throughout, basically thinning. So we wanted to know what is the zone of influence of living trees, basically on the soil. And how we did this was to actually make use of stable isotopes. And some of you may not be so familiar with what stable isotopes are, but basically all elements of interest in the environment, so carbon and nitrogen, have two stable forms. So in terms of carbon, we have 13C, which is heavier than the normal 12 carbon. So it has an extra neutron in its atom, and it exists in the atmosphere at very, very low concentrations. So the less than 1% of carbon is found as heavy, or 13C. And we can make use of that factor that we have this heavy isotope of carbon that's rare, because when plants photosynthesize or fix the atmospheric CO2 into sugars, plants discriminate against that heavy carbon. So they will preferentially fix 12 carbon. So we can use that as a signature to find out where the carbon from the tree is going in the soil. So I had a PhD student, Carolyn Churchland, who was looking at sampling soil in this 27 by 18 meter grid from this aggregated retention patch out into the clear cut. So she included half of the retention patch, and then going out into the cut area, you can see the flags. So we had flags every 2 meters in each direction, and she took samples looking at CO2 from the soil atmosphere, and also sampling the soil to look at microbial biomarkers. And what we can do is extract lipids from the soil that are characteristic of different microbes. And then we can measure the 12C to 13C ratio and use it, basically using that method. We can then determine where did those microbes get that carbon, because as I mentioned, when plants photosynthesize, they select against the heavy isotope of carbon. So any carbon coming out of those tree roots is depleted in 13C. So these are basically just heat maps showing you the signature of the gaseous CO2 that's coming out of the soil in these different areas. And what I want you to take home from this is that the CO2 being released in the retention patch and 3 meters out was depleted in 13C. So it had the signature of the carbon coming from the tree roots. And when we extracted the lipid from fungi from the soil, we can see that the fungal biomarkers are again depleted in 13C. So they have the same signature as the trees. So they're actually getting carbon from the trees. And this is within the zone of influence is about 6 meters out from where those living trees are. So we know these living trees are supporting soil organisms a few meters into that cut area, but then the lipids become more enriched in 13C, which shows the carbon is coming from this cut area. So what we wanted to do, although we can use this natural abundance of 13C, we wanted to see the actual flow of carbon from trees into microbes, but also small soil animals, so things like mites and enkitrade worms in the soil. So what we did was, with a colleague from the University of Cumbria in the UK, we labeled trees with a 99 atom percent enriched 13C. So basically glucose that was labeled purely with 13C, so that we could then follow that 13C from the tree out into the cut area. And again, we measured 13C in the CO2 that was being released at different distances, and in the roots, in the soil microbes and the soil fauna. And I'm showing you this illustration, this cartoon of what actually happened after we labeled the trees, shown in purple with this 13C glucose. So we had 13 CO2 being released at different distances from the plant. So that respired CO2 peaked in 13C after 2 weeks. We had dissolved organic carbon in the soil, also picking up the 13C label, and the signature biomarkers from the microorganisms we found peaked in 13C after 2 to 4 days, and this was 5 to 10 meters from the living trees. And then we found that mites and enkitrade worms, in turn, on days 4 and 6, were enriched in this 13C. So we knew they were eating the microbes that had eaten the carbon from the plant roots in this system. So what I wanted to say as a take-home message from this is that I'm showing some pictures here. This is just off the Egmont Road. So in Egmont now we have 3 lonely dugs in the area that they've been clear cutting with reserves. So clear-cut areas, clear-cut logging severs the lifeline of living roots in forests and devastates soil biodiversity and abundance. The clear-cut with reserves policy that's now dominant in BC forestry retains single patches of trees, like here on the right, on 3% of the harvested area, and it's not adequate to retain the soil organisms and their functions. You've seen from that evidence that living trees influence microbes, but the extent of that influence is really gone by 10 meters. So we need to adapt our forest harvesting practices to foster the life below ground and to make sure that we're retaining our soil carbon in these forests. So retaining patches of living trees sustains soil biodiversity within the patch and for a few meters out into the cut. But as I said, it's negligible by 10 meters. So trees need to be dispersed throughout the harvested area, as I've suggested here, with inter-tree distances no greater than 15 meters. So this works out to about 40 live trees per hectare. Okay, and I wanted to just finish by thanking the people that have contributed to the work I've presented. Again, the photographers Oliver and Nicole in Germany. And for those of you interested in learning more about this and our proposal for continuous root forestry, Cindy and I have just submitted a manuscript to Forest Ecology Management on this topic, and so I'll keep you posted on that. So thank you, and I'll take any questions. I guess I can stop sharing.
Yeah, if anybody has any questions, you can either put it in the chat or just put your hand up and then we'll.
Or you can email
there.
me. Yeah. Everybody's blown away.
Well, first, Sue, I just wanted to say that, you know, that was a fantastic presentation. And who knew that it was so colorful in the soil? Like, we look down at it and it's just brown.
I should have said those scanning electron micrographs, they're in black and white, so they've been color enhanced.
Okay, I wondered about that.
Yeah.
But it's still amazing to see all these creatures under there.
Yeah.
Okay, so there's a question in the chat here. Well, Maureen says fascinating, and then Jackie says, when nursery soil is sterilized, do all these organisms die?
Yeah, so if they're gamma irradiated, they will. Some of the spore-forming organisms, some bacteria like bacilli that form spores that are resistant to a lot of treatments. But in terms of nurseries, they like with the seedlings that are grown, they won't necessarily sterilize those, and the seedlings become colonized by mycorrhizal fungi, but they're usually replaced from what was present in the nursery to what's out at the site once the seedlings are planted. But if you heat sterilize soil, you will kill some of those organisms. So I think a lot of sterilization is done to kill off pathogens. If you're continually growing plants in the same soil, you'll get a buildup of pathogens. But you could, for example, use a slurry from a fresh soil and use that to re-inoculate if you've sterilized your soil to get rid of pathogens. But yeah, we do know, for example, I didn't mention the work we've done looking at soil animals in these retention areas and at distance from trees. So we found, for example, that Collembola pauropods mites, they all declined with distance from the living trees. And another interesting thing was that before harvest, you had this great diversity of different mite species. But after harvest, the mite community became dominated by one particular species. And, you know, what does that mean for the function of that ecosystem? You know, why is that particular organism mite dominating? But thinking if they have different functions, you know, we don't want to, you know, we hypothesize that if we have a lot of diversity, we're going to have some redundancy in the soil, so that if we lose some organisms, maybe some can take over that role. But because we don't know all the functions of the organisms, we don't know what are the consequences for losing some of them.
Ray, yeah, Ray has a question.
Is that me, Carol?
Yeah.
Yes.
Okay. Hi. Yes. Thanks, Sue, for a really fabulous presentation, and it was worth the wait. So technical difficulties. I just had a question about, before we started watching this, there was something that came up, I think it was CBC News, about Canada ranking 58 out of 63 countries in terms of its climate change policies or performance. And I just wondered, and I think you touched on this, what role do these soil organisms have in carbon retention and ultimately in the release of CO2 to the atmosphere?
Yeah, so that we now know that actually soil microbes are the main bulk of the stored carbon in soil. So the soil organic matters, these microbes are eating the carbon, the labile carbon from the plant roots. They're then growing themselves, and then they're the basis of what we call the mineral associated organic matter in soil. And that's really recalcitrant. But if we're cutting off their carbon, their food source, we're reducing the carbon storage in soil. So not only have we got less trees fixing carbon and less carbon going to the soil, then the microbes that eat that carbon are less. So we're going to get less carbon stored in our soils. So we know, for example, after we've harvested, that the storage of carbon in those forests doesn't recover to pre-harvest levels for about 20 years. So yeah, I mean, they do respire CO2 in the same way we do, but in terms of, the other greenhouse gases I mentioned, methane, so some of the microbes are important. There's certain groups of what we call methanotrophs that can take up methane and convert it to CO2, which is less potent a greenhouse gas than methane. So our forest soils are a fairly important methane sink.
Okay, there was another question in the chat by Tamara. Do you know if the Douglas fir cone mushrooms inoculate on the tree or once they hit the ground? The Strobilurus chulicitus. Did I say that right?
This is a.
I think it's the little white mushrooms that are on the Douglas fir cones that we see around this time of year. I think that's what.
Oh, the
okay, she's nodding.
Decomposers. Yes, yeah. So whether they're actually on the cones before they come down.
Right.
Yeah, because that's what happens with, a lot of like the maple leaves, the big leaf maple as well. So they're already infected with fungi, the black dots on them. So they're starting to decompose before they come onto the floor. And then you have like the millipedes eating them. But yeah, and a lot of like the spores are all in the atmosphere.
All right, I don't see any more questions in the chat. Last chance for a question.
Yeah, question here.
Oh, here's John.
John.
Hi, Sue.
Hi.
Yeah, just wondering who's listening to you other than us? Are this knowledge that you're gaining, is how long before it's likely to affect forestry practices?
Oh, well, I'm hoping like we've got this paper we've submitted, but then the politicians don't want to hear. They want 10 minutes. You've got 10 minutes to tell them why they should be interested and what they're going to do to change. So I mean, with the logging around Egmont, I wrote to all the ministers about that and why aren't we doing selective harvesting as they're doing in Scandinavia. So this leaving basically dispersed retention. And like the Minister of Forests at the time, the Minister of Environment wouldn't touch it because they don't want to step on each other's portfolios. So that was George Heyman. So yes, I don't know. I think if like we can have more popular articles, like I've had quite a few letters and books sent to me since the National Geographic article came out. And I sent that to one of the ministers as well, and he just said thank you. So,
Right.
We need to send them the link to this presentation and make them sit down and watch it. I actually had a question.
I think public pressure and
Yeah, good. I was wondering, did you say that once the soil, like once they clear cut and the soil kind of gets void of the biodiversity, did you say it takes 20 years to recover back to?
so the soil carbon,
Okay.
yeah. So the organisms, so they can come in if you have the living tree still present, they'll be retained, and so they can move out from there. But if we're thinking about like soil mites, I think it's been estimated in a lifetime, a mite can move about half a bowling lane in length. So it's not very far. It's not going to go very far.
Right. All right.
But
Well,
I know when they have, I guess the next presentation next year is in Mount Polly. So there was a student that was on her committee and she was looking at restoration of forests in that area after the spill. And so she did use like an she took a soil inoculum from the forest that was okay at the side and used that as an inoculum for seedlings that were put in, and that seemed to work pretty well.
Ray Kostachuk here again. I'm actually giving that presentation on Mount
Oh,
Paul.
right.
Okay. Yeah.
So this was Katie McMahon.
No, any way I can get the reference to the publication?
Yeah, she has a couple of papers now. I'll send them to you. Yeah.
Okay. Thank you.
Yeah,